Upgrade of design standards and redesign of bioretention systems, presented by the Environmental Research Group at the University of Tennessee at Chattanooga.
An alternative to the traditional approach to mitigate urban stormwater issues is the use of green infrastructure, such as bioretention systems, to control runoff. If properly designed, installed, and maintained, bioretention systems capture and infiltrate stormwater while removing pollutants, reducing flow rates and runoff volumes, providing species biodiversity, and improving the socioeconomics in the surrounding area. However, the lack of design criteria and maintenance standards is a major impediment to implementation and financing. Towards developing optimized design standards for bioretention design, implementation, and maintenance, we used existing bioretention at Warner Park in Chattanooga, Tennessee as a case study. More specifically, as part of this project, we developed 1. Sampling devices for improved influent and effluent sampling, 2. Design standards that are sensitive to drainage area land use, 3. A comprehensive checklist to assist with right design and proper field implementation, and 4. A detailed quantification of the social, economic, and environmental benefits bioretention can bring.
Bioretention is challenging to design and maintain. To determine if a site should be retrofit for bioretention or if it is cost prohibitive to install, we developed a site assessment and design checklist. It can be used by engineers, contractors, state or municipal government, or property owners. Our checklist is initially a planning tool that helps to determine if the site is applicable or feasible. It then looks at the application, site requirements, recommended analysis and design programs, physical requirements of the cells, underdrain and sampling tube requirements, planting guidance, and maintenance. It also addresses the benefits including social, economic, and environmental. This tool can be used by anyone needing to lower their stormwater fees or reduce pollution impacts to nearby streams as determined by the EPA.
The flowchart shown below shows the process that led up to the development of our sampling weir. The first picture demonstrates a sampling method with a 1-liter Nalgene bottle to store water and a small cup to collect grab samples. That small cup has to be placed on the parking lot while there is sheet flow, and as it collects the sheet flow, sediment is being picked up, creating a biased sample. As we realized that collecting sheet flow with that method proved difficult and our samples were contaminated, other sampling techniques were explored. A portable weir was designed to increase inflow depth, and the models are shown below along with the final design on the right-hand side. Conducting water quality monitoring is necessary to assess bioretention performance. Influent and effluent sampling is needed to evaluate bioretention design and maintenance. During the case study, one challenge was identified with collecting influent samples for sheet flow. To overcome this, we designed a portable broad-crusted weir to collect unbiased sheet flow samples. Without the weir, samples were biased due to collecting sediments off of the ground. The initial design was designed to fit over the curb stops at the site. The weir with open slots was designed to hold weight, such as water, to prevent sheet flow from going underneath it. The first design's applicability is limited to testing areas with curb stops, while the weir with open slots is applicable for any site with sheet flow on an impervious surface, making it more portable and the better option. This design creates enough water depth for accurate sampling while avoiding unnecessary flooding, as it allows runoff to overtop in the bioretention if the design sampling height is exceeded.
The approach that we took to produce design recommendations was to run alternative designs in a computer model. We used CLASSIC, which stands for Community-Enabled Lifecycle Analysis of Stormwater Infrastructure Costs. CLASSIC is a free web-based software tool prepared by Colorado State University's One Water Solution Institute, and it produces hydrologic results along with cost estimates and co-benefits. The results of the hydrologic performance analysis for 3 of the scenarios are shown here. The baseline model was set up to closely mirror the conditions at the site as they are now. The residential model was set up with less impervious surface area, 20% compared to 93%, a smaller total bioretention area, no underdrain, and less media depth, 18 inches compared to 30 inches. The deeper media model was designed with all the same design parameters as the baseline, but the media depth was increased from 30 inches to 36 inches. The reports of the model indicate that the deeper media design was the most efficient at removing pollutant load and also produced an increased rate of infiltration and a reduced rate of runoff compared to the baseline model. These results led to the conclusion that increasing the media depth increases the performance of bioretention systems.
To better understand bioretention, we studied the bioretention cells in the parking lot at Warner Park Zoo in Chattanooga, Tennessee. The project site is 2.59 acres, and 79% is impervious. This means most of the water on our site is being diverted into the storm drains and being sent to the combined sewers. Instead of sending the water off-site, the bioretention is capturing the runoff, thus allowing it to be naturally cleaned, then infiltrated, which recharges the base flow. This bioretention system is made of over 90 different plant species, mostly all native, and the soil is an engineered design. To get comparison information on our site, we collected samples of the runoff and ran them in our lab to test for pH, conductivity, dissolved oxygen, turbidity, total suspended solids, and total dissolved solids. While collecting samples, we realized that there was high amounts of sediment and also realized there should be an easier way to collect samples that would increase the sheet flow water depth.
One consideration for our bioretention project is the implementation of our retrofit design at our case study area of Warner Park. All cost derived for this analysis comes from the Water Environment Research Foundation's BMP and LID Whole Life Calculator. Our total project cost over the 50-year period came out to be, in present worth value, $135,168. Of this amount, our capital cost or implementation cost came out to be $106,835, while maintenance over a 50-year period equated to $28,333. Part of our consideration was also to see what kind of revenue we could quantify from bioretentions. Two things that we were able to quantify for consideration was water quality fee discounts following the City of Chattanooga's incentives manual, along with a best case scenario of water harvesting. These two benefits put together equated to $1,678 in annual savings, but unfortunately our annual maintenance costs came out to be roughly $3,390. Given these facts, our project is considered non-refundable based on the benefits we were able to quantify. It doesn't mean that bioretentions are completely a loss in profit, given that there are several yet to be quantified benefits of bioretention environmentally and socially